Welcome to the deep dive. We're here to cut through the noise and get straight to the core knowledge right from the source. And today, wow, we are diving into something that sounds almost, well, counterintuitive. The idea that parts of our DNA, stuff scientists literally call junk, might actually hold the key to treating some really tough cancers.
It's a fascinating premise.
Isn't it? So our mission today is to really dig into this groundbreaking work from King's College London. They've figured out a way to sort of flip the script on two difficult blood cancers, myelodysplastic syndrome, MDS, and chronic lymphocytic leukemia, CLL.
And the crucial part here, I think, is that it's not about inventing some brand new drug from scratch. It's about finding a hidden weakness, a vulnerability within the cancer cell itself, and then using drugs we already have to exploit it.
Right, repurposing.
Yeah.
And that's so important because, as you know, for many people facing MDS and CLL, the usual treatments just, well, they don't always work long term.
Exactly. The options can become really limited. These diseases desperately need new approaches, new angles of attack. What we're talking about could be a real paradigm shift for how we handle certain cancers that have been very hard to treat.
Okay, so let's unpack that core mystery then. Why aren't the conventional treatments working so well for these specific cancers? What's fundamentally different here?
Well, it really starts at the genetic level. When you look closely at MDS and CLL, you often see these major mutations, these failures in specific genes. We're talking about ASXL1 and EZH2.
ASXL1 and EZH2. Okay, so what do these genes normally do? I think you described them before as kind of like master circuit breakers.
That's a pretty good way to put it, yeah. They're key regulators. Their main job is to control other genes, basically telling them when to turn on, when to turn off. This controls normal stuff, like how cells grow and divide.
So they keep everything orderly.
Precisely. But when these genes get mutated, it's like, well, it's like the main control panel is broken. You lose that regulation, and that leads to the out-of-control cell growth that, defines cancer.
Okay, but here's the challenge, right? For the drug developers.
Yes. So standard cancer therapies, the targeted ones anyway, usually work by finding a specific harmful protein that the faulty gene is making too much of, or that's malfunctioning. And then the drug is designed to block that protein. It's like putting a stopper in a faulty machine.
Like plugging that leaky pipe, as we said.
Yeah.
But the twist here with ASXL1 and EZH2 mutations is that sometimes there's no pipe.
Exactly right. It's a bit more complex, but sometimes the mutation means the gene completely stops working, a loss of function. So that harmful protein, the one you'd normally target, it often just isn't there. It's absent.
Yeah.
So if your drug's job is to block something and that something doesn't exist, well, there's literally nothing for the drug to inhibit.
Wow. Okay, so that's a major roadblock, a dead end for that kind of therapy.
It really is. It means we have to look somewhere else entirely for a weakness.
Which raises the fascinating question.
Yeah.
If the obvious targets aren't there, where do you look? Sounds
Yeah.
like some serious molecular detective work was needed.
Absolutely. And since targeting that mutated regulatory protein pathway was, well, a bust, the researchers had to think differently. They started looking into the less understood parts of the genome, the stuff often dismissed as, well, junk DNA.
Okay, here's where it gets really interesting. This junk DNA, we're talking about transposable elements, right?
Yeah.
TEFs. Give us a sense of scale. How much of our DNA are we actually talking about here?
It's honestly staggering when you think about it. Nearly half, something like 45 to 50% of all our DNA is made up of these transposable elements.
Yeah, wow.
Yeah, they're essentially these little segments of DNA that have the ability to copy themselves and then jump around to different spots in the genome. For a long, long time, most scientists just thought they were evolutionary leftovers.
Yeah.
Non-functional clutter. Just molecular debris that built up over millions of years. Irrelevant.
But now this debris, this supposed clutter, turns out to be the Achilles heel for these specific cancer cells.
Precisely. That's the core of the discovery from the King's College team. They found that in these cancers, the ones with the broken ASXL1 and EZH2 genes, those faulty circuit breakers, these transposable elements, suddenly become abnormally active. They start moving around.
So the regulators are down, and this old stuff starts waking up.
Kind of like that, yeah. You could think of the Ts as maybe old, dormant code, biological malware that was normally kept suppressed. But now the system administrators, ASXL1 and EZH2, are offline, and this old code starts running wild.
Okay, and what happens when they run wild?
Well, when these TEs start copying themselves and inserting into new places in the DNA, they cause chaos. They physically break the DNA strands. This activity puts enormous stress on the cancer cell. It's constantly damaging the cell's own DNA structure.
Ah, so the junk is actively harmful in this context.
Critically harmful. And this constant damage, the strain caused by the TEs themselves, that's the surprising vulnerability. It's a level of internal chaos the cancer cell struggles to cope with.
That's incredible. The cell's own, supposedly useless baggage is pushing it towards breaking point because its main controls are already broken. What irony.
It really is molecular irony, as you put it.
So, okay, we've identified the vulnerability, the chaos caused by hyperactive TEs. Now, how do you exploit that? This must be where the PRP inhibitors come back into the picture.
Exactly. This is the clever repurposing part. PRP inhibitors aren't new drugs. They're already used in oncology.
Right, I've heard of them, often with BRCA mutations like in breast cancer.
That's their usual role, yes. In those cases, like BRCA-related cancers, the cancer cells already have a hard time repairing certain kinds of DNA damage. PRP proteins are part of another repair system. So using a PRP inhibitor basically blocks one of the cell's remaining repair pathways. The idea is to prevent the cancer cell from fixing any DNA damage, causing it to accumulate until the cell dies.
Okay, so that's the standard job. How is it different here with the TEs?
It's much more specific. Remember, the Ts are constantly jumping around and causing new breaks in the DNA, physical breaks. PRP proteins, you can think of them like an emergency repair crew, they rush to the site of these breaks to stabilize the situation and kick off the repair process. They're essential for fixing the specific damage the TEs are causing.
Ah, I see. So the TEs are like vandals constantly breaking things.
Yes.
And the PRP proteins are the crew trying to frantically patch everything up.
That's a great analogy. So if you bring in a PRP inhibitor drug in this specific situation,
You're not just stopping general repairs, you're specifically stopping the repair of the damage caused by the TEs. You're blocking the repair crew right where the vandals are most active.
you've got it. You're essentially sabotaging the cell's attempt to clean up the mess made by its own hyperactive junk DNA. Because the TEs are already causing so much intense ongoing damage, blocking the PRP repair pathway means the damage accumulates incredibly quickly, much faster than in the standard PRP inhibitor scenario. It pushes the cancer cell past its breaking point, leading to catastrophic failure and cell death. We're basically turning the cell's internal chaos against itself,
Yeah.
very directly.
Okay, but help me understand one thing. If the TEs are already causing DNA damage and chaos, isn't that bad enough? Why is blocking the repair better than just letting the TEs cause havoc? Doesn't more damage just mean potentially more mutations?
That's a really crucial point. It's about the type and speed of the consequence. The ongoing damage from TEs alone causes genomic instability? Yes. Mutations accumulate, and eventually this can contribute to the cancer progressing and becoming more aggressive, ultimately harming the patient. It's a chronic simmering problem for the cell.
Yeah.
What the PRP inhibitor does is take that chronic instability and turn it into an acute crisis. It accelerates the accumulation of a very specific type of damage DNA strand breaks to an overwhelming level very quickly. It pushes the cell from a state of struggling with damage to catastrophic system failure. It forces the cell over its lethal threshold almost immediately, preventing it from adapting or surviving that damage long enough to cause more trouble down the line.
Got it. So it converts a slow burn into an immediate blow-up.
A very effective blow-up in this case.
That specificity is key then. But how did the researchers prove this? How did they confirm it was really the TEs causing the problem that the PRP inhibitors were exploiting, and not just some general stress response?
This is the really elegant part of the research, the confirmation step. They used another type of drug alongside the PRP inhibitors. These were reverse transcriptase inhibitors, or RTIs. You might know them from HIV treatment, actually. In this context, RTIs have specific effect. They stop the transposable elements from being able to copy themselves. They essentially block the TEs from jumping around. They stop the vandals.
Okay, so they put the brakes on the TE activity itself.
Exactly. And here's the clincher. When they treated the cancer cells with both the PRP inhibitor and the RTI, the killing effect of the PRP inhibitor disappeared.
Wow. So if you stop the TEs from making the mess,
Then blocking the cleanup crew, the PRP proteins suddenly doesn't do much. The PRP inhibitor loses its power.
that's definitive then.
It really is. It proved, pretty much beyond doubt, that the whole therapeutic strategy using PRP inhibitors to kill these cells was entirely dependent on the TEs being hyperactive and causing that initial damage. They nailed down this unique TE-specific vulnerability that could be targeted with drugs we already have.
That is just Fantastic science. Professor Chiwei Eric. So at King's College, London really summed it up, didn't he? Saying this gives real tangible hope by turning, quote, useless DNA into a powerful target for treatment.
Did a perfect summary.
But let's think bigger picture now. This research focused on MDS and CLL, linked to those ASXL1 and EZH2 mutations. What about other cancers? Does this principle apply more broadly?
That's the exciting implication. The underlying principle here is about what happens when these key regulatory genes fail. So the findings strongly suggest that other types of cancer, potentially many others, that also have mutations in ASXL1 or EZH2 or perhaps similar regulatory genes.
I have the same hidden vulnerability, the same hyperactive TEs.
It's highly likely, yes. Cancers where maybe conventional drugs that target proteins also hit that same dead end because the protein is lost. These cancers might also rely on PRP to clean up the mess from their own rogue TEs.
So this research isn't just a specific fix for 2 blood cancers. It could be a blueprint.
It absolutely could be. It potentially opens the door to using PRP inhibitors in a whole range of cancers where they aren't currently considered effective.
Potentially unlocking a massive second life for these drugs. Talk about efficient science finding new uses for existing tools.
Exactly. Getting more mileage out of the drugs we have is always a huge plus.
What an incredible journey this deep dive has been.
Yeah.
We started with this frustrating situation. Key genes like ASXL1 and EZH2 fail, leaving standard therapies kind of stumped. Then the researchers pivot, look into the genomic dark matter, the so-called junk, and realize that's where the real vulnerability lies, these hyperactive transposable elements.
Transactive culprits.
And now we see a clear path to exploiting that flaw using existing PRP inhibitors, confirmed by that clever RTI experiment. It's just brilliant.
It really forces us to rethink what we label as junk in biology, doesn't it? If nearly half our DNA, stuff dismissed for decades, actually holds a critical switch for treating devastating diseases like MDS and CLL, it makes you wonder. And maybe this is the final thought for you, the listener, to mull over. If this principle holds, what other parts of our biology, tiny RNA fragments, maybe ancient viral DNA embedded in our genome, things we currently filter out as just background noise, what other overlooked junk might actually hold profound answers, powerful solutions for diseases we still think are incurable? What else are we missing?